Hi, my name is Zach Duff, and I'm going to discuss the paper Calpain 6 Deficiency Promotes Skeletal Muscle Development and Regeneration. The reason why I chose this paper was because I've always had an interest in the muscular system, and I wanted to see if there was a natural protein that either enhanced or suppressed muscle growth. Here are the original authors of the paper. So before we begin, we need to get a little background on calpains. A calpain is a protein that belongs to the family of the calcium-dependent cytosine proteases. In mammals, there are known to be 15 calpains present, and they are thought to play roles in cell migration, apoptosis, platelet formation, and myoblast fusion. This experiment specifically focused on calpain 6, which is thought to play a role in embryogenesis, and it's only expressed on the X chromosome. Calpain 6 is also only present in embryonic muscles in the placenta. Previous experiments show that calpain 6 plays a role in microtubule dynamics and actin reorganization, but the physiological functions of calpain 6 are still very much unknown. This experiment hoped to shine a little light on the physiological roles of calpain 6 by examining them in vivo using mice. This is a schematic diagram of what they hope to accomplish to silence the calpain 6 protein. Up top, we have the wild type allele. Here's the 5' end and the 3' end. And here's exon 1, exon 2, and exon 3. Right here in the middle, we see what they wanted to insert to disrupt the gene locus, and that's the LAC-Z cassette. The goal was to insert the LAC-Z cassette in between exon 2 and exon 3. After they manually inserted the LAC-Z cassette between exon 2 and exon 3, they wanted to breed the mice to obtain fully deficient calpain 6 mice. So they bred a calpain 6 heterozygous female, because again, the calpain 6 protein is on X chromosomes, and they bred a fully deficient calpain 6 male, since males only have one X chromosome, its X chromosome would be calpain 6 deficient. What they hoped to obtain was again a fully calpain 6 deficient male and a homologous calpain 6 deficient female. To confirm this, they used Western blot because Western blot will identify specific proteins in a sample. So here's one of the Western blots. This one was of a whole embryo. The goal was to not see a band at 76 kD. A band present at 76 kD would indicate the presence of the calpain 6 protein. Here's the embryonic days. Again, calpain 6 is only expressed during embryogenesis. Here we see the genotypes. Wild type is where we expected to see the calpain 6 band, and we see that in all 3 samples. The hemi is where we did not want to see the calpain 6 band, and again, in all 3 samples, we did not see a band. Here's a Western blot of tongue muscle cells. Again, the band would have appeared at 76 kD, and the genotypes are the same as well as the embryonic days. Something different that happened with this Western blot was the embryonic days were later in the cycle, and again, since calpain 6 is only expressed during embryogenesis, as embryogenesis came to an end at 18.5 days, the calpain 6 expressivity was dying down. Here's a final Western blot of limb buds. Again, it's the same scenario. At 76 kD, we expected to not see a band. In the heterozygous, we expected to see a band in both mice, male or female. Whereas in the homologous, for the female, we expected not to see a band. And then for the hemi and the male, we expected not to see a band, which was confirmed. So here's the first piece of results that they attained in regards to muscle fiber size. The white bar indicates calpain 6 mice that had the calpain 6 protein, whereas the black bar indicates the calpain 6 deficient mice. As you can see, the calpain 6 deficient mice had a much larger muscle fiber diameter throughout embryogenesis than the calpain 6 mice. This table indicates the number of nuclei per cell throughout embryogenesis up until day 9. Again, the white column indicates the calpain 6 mouse, whereas the black column indicates the calpain 6 deficient mice. each day, you see that the Calpain-6 deficient mice had an increased number of nuclei compared to the Calpain-6 mice. The increased number of nuclei indicates that muscle will regenerate faster and will have better development than those with lower nuclei numbers. This is a diagram of individual muscle fibers. The control, it should be fairly similar, as well as the 4-day, there's not much difference. But over here on the left, in the 7-day, you can see the Calpain-6 mice that had the Calpain-6 protein present. And over here on the right, you see the Calpain-6 deficient mice. And at day 7, we saw a noticeably bigger difference between the Calpain-6 deficient mice and the Calpain-6 mice. That the Calpain-6 deficient mice had larger muscle fibers. This is slides of tongue muscles from the Calpain-6 mice. Over here in the A box is the mice with the Calpain-6 protein, and the B box is the mice with the Calpain-6 deficiency. I want to point your attention to right in this area where the square box is. These are the striations of the muscle cells, and you can see between A and B that the Calpain-6 deficient mice had increased size and striations, therefore again indicating that a Calpain-6 deficiency will promote skeletal muscle growth. A couple pieces of results that they didn't really have pictures for, but they wanted to discuss significantly was a beta-galactosidase staining that they performed, and this led them to more results that they were not expecting, which I will discuss in the next slide. Another piece of evidence that they found was that the Calpain-6 deficient mice, at the end of the embryogenesis, the embryos weighed more and had a higher muscle percentage, which again fell into the results that a Calpain-6 deficiency will promote skeletal muscle growth and regeneration. So all of these results gave the experimenters a conclusion, and like I've been saying, the whole presentation is that Calpain-6 deficiency will promote skeletal muscle growth. So therefore, that must mean that Calpain-6 acts as a suppressor to skeletal muscle development and regeneration. And it's thought to do this by modifying cell division, cell growth, and cell migration. Now, in regards to the beta-galactosidase staining that I discussed, they took whole embryos at the end of the embryogenesis period and stained them with the beta-galactosidase. And what they found was that Calpain-6 is not only expressed in skeletal muscles, but it's also expressed in the cartilage, the somites, and the myocardium. Now, the researchers were not expecting this, and what the conclusion they came from was, and again, this will take more experiments to conclude, is that Calpain-6 acts to protect the mother. It prohibits the embryo from going too large in the mother's body, which in turn can harm the mother. So in the future, there's still a lot to learn about Calpains, but they want to explore the mechanism that Calpain-6 uses to suppress skeletal muscle growth, as well as suppress the growth of the somites in the cartilage and the myocardium. And they also want to look at functions of the other 14 Calpains to determine if they have any other life-threatening functions that Calpain-6 has. Like I said, Calpain-6 prohibits overgrowth of the embryo to protect the mother. So they're curious to see if the other Calpains will play a role, just like Calpain-6 does. Here are the resources.